Pig xenotransplantation. Yeah, that's a mouthful. While it sounds complicated, the concept is actually easy to understand. Pig xenotransplantation is the term for using genetically modified pig organs as replacements for human organs in organ transplant surgeries. But why is this important? Humans have been transplanting organs for decades, but there is a much higher demand for human organs than how many are actually available. According to the Health Resources and Services Administration, 17 people in the US die each day waiting for an organ transplant. To counter this, pig organs have been coming into the light as a possible alternative to the organ shortage. But you might be thinking, okay, why pigs? Pigs reproduce very quickly, and they mature at a very quick rate, which means there would be a greater supply of organs, not just as a solution to the shortage, but as a solution to providing an inexpensive alternative to what is an extremely costly procedure. Surgical procedures using pig tissue, including skin grafts, have also been successful in the past, providing hope for organ transplants in the future. But how does it work? Scientists extract DNA, the body's instruction for how to grow, from pig eggs, and they make necessary genetic modifications, including ensuring that the organs won't grow to be too large and the human host won't reject them once they're transplanted. The DNA is then reinserted into the pig eggs, and once they grow to be embryos, surgically implanted into mama pigs. These genetic modifications also mean that the piglets born are genetically identical clones, and then companies raise these pigs in sterile farm facilities to prevent contamination. Their organs, once mature, could hypothetically be used in organ transplants. Now let's dive into the nitty-gritty. What's one gene edit made to this pig DNA, and why is it important? PERVs. No, it's not what you think. One major issue with using pig organs is the possibility of spreading viruses into the human system. In the pig genome, these are called porcine endogenous retroviruses, or PERVs, and they're a family of viruses that could potentially transmit to the human host, and they could be deadly. In fact, PERVs are very similar to leukemia, so this would be a serious problem. To counter this, scientists have used a gene editing tool called CRISPR-Cas9 to target 25 different sites along the pig genome, and they've successfully deactivated them. It's pretty nifty. Beyond PERVs, there are several concerns, but most boil down to that there isn't enough certainty that using pig organs would be safe and that the human host would accept them. So far, everyone who has had a pig organ transplant has passed away, though often from other health concerns. However, pig organs have been successfully transplanted into human hosts. Also, there are ethical dilemmas, such as growing pigs solely for organ harvesting. So while it's true that testing, research, and careful consideration definitely needs to be done, pig organs provide an enormous hope for ending the organ shortage and saving lives, and scientific progress continues to be made. Pigs, our lives are in your hooves.